agora vamos passar à apresentação da Fermentis Sameca sobre o Prise De Mousse, com a primeira etapa de preparação de leveduras, mas abreviada. Quem vai lecionar ou orar nesta palestra vai ser o Richard Marchal, que é doutor Richard Marchal, que é doutorado em Bioquímica e Biologia Molecular, entre outras conquistas. Chama ao palco Richard Marchal.
Okay. Good morning, everybody. Thank you, Inoculum, for this nice organization. And very happy to be once again with you in Portugal to present our study about the selection of a new yeast strain and how it's possible to reduce the time necessary to prepare the yeast starter to achieve the Prise De Mousse. So the talk will be in 7 points. The first point, a small introduction. Points 2 to 4, how it was possible to select a new strain. Point 5, what happens during the starter preparation. Point 6, we propose a new protocol. And for the last point, we will compare the classical protocol and the new shortened protocol. Then just to give a small definition of the expression and the words we will use, we say Prise De Mousse. In fact, it's a French expression. It's the secondary fermentation that will occur generally in the bottle. It can also be in a closed tank for the Charma method. And after we begin with a base wine, and after the secondary fermentation, we have a sparkling wine or effervescent wine. And to achieve this second fermentation, we need a starter, a yeast starter called pied de cuve, pied de cuba. And for this, the quality of the starter is essential to achieve correctly the kinetics and the fermentation. Traditionally, it's made in 3 days, the classical protocol, in 2 steps, and we will try to see if it's possible to propose a reduced, a shortened protocol, reduced by 50%. It means 36 hours or 1 day and a half, and you will see that it's very important in practically in the winery. And also to propose this new protocol, it's necessary to have a close-up on what happens, especially on the nitrogen and sugar needs for the yeast. And the second point, this study is a partnership, a collaboration between Fermentis, Le SAF, for the industrial part, and also the Institut Français du Vin, University of Reims, and where I live, I work, from the north of France in the cool climate region. It was cool, not so much now. And also the University of Haute-Alsace on the east of France. Then first step, the selection of the yeast strain. We begin with a, I will change. Okay, I will see better. Sorry. Then we work with a Pinot Meunier, a blend with Pinot Meunier and Pinot Noir. We are in the north, and you know we produce the champagne with essentially black grapeberries. And then we use for this study always the heart of the flesh to avoid the too high level of pink color. You can see that essentially we have a pH for all these studies. We have a pH very close to 3. Sometimes a bit lower, a bit higher, but it's always around 3. And it's not so much, but it's very nice to have to keep the freshness. First step, we have the comparison of 5 strains, and one of them will be called after the SPK05. This is the selected yeast strain. We can see that all of them are efficient. We can achieve completely the alcoholic fermentation. This is the first alcoholic fermentation. And the SPK05 is quicker, is very quick, and we can see directly for the hydration step, we have the production of the foam that is very important, and quickly we have a very nice pied cuve. If we have a look on the wine, the current analysis of the wine after the alcoholic fermentation, we can see here, for example, we are always around 10 grams per liter in equivalent tartaric acid. It's not so high, sometimes it's a bit higher, because we can have until 10 grams in H2SO4 than when you have less sun, and you keep in mind we are in the north. We can see here we have a lower SO2 content. This is an interesting point, reduction. A small, smaller quantity for the malic acid. It's very, the difference is not so much, but we can see that it's always the same. Then to summarize, we have less SO2. This is interesting point. Less malic acid, and of course a lower total acidity. Not so much, but lower. This is the first step, the alcoholic fermentation in the tank, and the second step is the prise de mousse, and the secondary fermentation in the bottle, the PDM. Well, for the mousse analysis, you can see it's pretty the same. The pH is 3.03. It seems to be low, but it's really the current value we can obtain. We can see after the alcoholic fermentation, the first alcoholic fermentation, a lower SO2 quantity, and as for the previous example, we have a less titrable acidity, a bit lower. We have here the difference of 0.4 grams per liter. Then we have no significant differences between the different strains. If we follow with the afrometer, the permanent afrometer, we follow the prise de mousse and the pressure in the bottle, we can see that finally for the three strains compared, we have a pretty complete kinetics. It's true that for the reference 2, it's lower. It will take more time. Finally, the prise de mousse was complete, but with probably 1 month if you compare with the SPK05. Then a complete and quick secondary fermentation in the bottle. This is an important point, and we are the complete consumption of the sugar. After 1 year in the bottle, a bit more than 1 year, we have tested the different wines obtained with the 3 strains, and we can see that for all of them, we have the qualities, and for the SPK05, it's true that we have a very clean, very bright, and very fresh nose. It's a very nice wine, obtained in good conditions. Of course, for the reference 2 and 4, the wines also presented some qualities, but for the reference 2, we have seen that these strains was eliminated because the longer time for the prise de mousse in the bottle. Then another example here, you can see for the pH, it's always pretty the same, around 3, and we have the alcoholic content that is sufficient. We can compare here the prise de mousse, secondary fermentation. In the bottle, we have here just 2 strains, the SPK05 and a reference of another strain. We can see that it's pretty the same finally, and we see that sometimes it's a bit quicker, sometimes a bit slower, but if we compare, there is no significant difference, and more than all, we can achieve correctly the prise de mousse in the bottle. This is the main point. We can consume all of the sugar introduced, and at the beginning, I think it was 20 or 22 grams per liter. That is a classical concentration. Then to summarize the characteristic of the wines obtained, then with the SPK05 strain, for the first step is the alcoholic fermentation in the tank. We have a quick fermentation. It's easy to rehydrate and to have a nice starter. Then we have wines very freshness, a lot of freshness, and very bright, very clean wines. It's the same for the after the second fermentation in the bottle, 1 year, but we have continued the testing after 2 years. It was pretty the same, more evolution, of course, but we see that we have a complete and relatively fast fermentation, no residual sugars, and also a very pleasant nose, freshness. That is very important because we need to have these aspects to integrate the acidity of the wines, the base wine produced in the northern climate. To summarize here the technical characteristics of the wine, first, the very high level of implantation. It's a very important characteristic, also low volatile acidity, high resistance to low pH, and in many cases the pH is lower if we work with the heart of the flesh of the grapeberry. At the beginning of the press cycle, we have the pH in many cases lower than 3. Then it's important to have a yeast capable to achieve in good conditions the fermentations. And also we have a very low SO2 production and lower needs in nitrogen or DAP, diammonium phosphate. Then if we have a look on the starter, the cuve, we will try to follow the sugar and also the DAP, the nitrogen needs. It's very important to achieve correctly the fermentation. Okay, I will show you all the slide at the same time. Okay, this is the classical protocol we use in Champagne. Then, in fact, we have a protocol in 3 days and 2 steps. The first step is always the same. It's dry yeast, DAP, diammonium phosphate, and sugar in water, and generally it's 1 day. And we need to decrease the density. After we begin here in the example, we have 5 hectoliters, and this is the quantity necessary to inoculate and for the tirage of 1,000 hectoliters. Second step, we have 25 hectoliters more. We add once again DAP, sugar, and wine for the acclimatization of the yeast, and this is a total of 30 hectoliters for the inoculation at 3% volume for 1,000 hectoliters. This protocol lasts 3 days, and we need to follow correctly the evolution and the changes in nitrogen content, because it's very essential if you want to achieve correctly, to prepare correctly the starter and to achieve correctly the fermentation. Then we have tried to see if it was possible to reduce or to increase the nitrogen content and the effect on the sugar consumption. Then we will see directly on the right the graph for the sugar, and we see that there is a small difference in the sugar consumption. After 17 hours, we see a decrease by approximately 50%. Why 17 hours? Because if you begin, I will say practically in the winery, if you begin Monday in the afternoon, you can begin the second step the following day in the morning. And for the DAP, because we have just DAP for the first phase, we can see that finally the aspect of the curve is always the same. They are parallel. The higher the DAP content at the beginning of the starter, the higher the content after 17 hours. Then in this case, we have no nitrogen deficiency, and we have to pay attention because it's very important, a very important parameter. So to summarize, we have seen that finally we have a strain selected after 4 years of trials, and with different steps. We have begun with 4 strains, and finally we have selected 1 strain, SPK05. Then the question is, we have seen that finally we don't use all of the sugar, we don't use all of the nitrogen, and we see that the starter continues in good conditions. So why not to reduce the time necessary to prepare the starter? At the beginning, 3 days is the classical protocol used in many countries, in many regions, and why not to reduce, and why not to be capable to propose a starter prepared in 1.5 days? It means 36 hours. You begin on Monday in the afternoon, and you are capable to begin the tirage on Wednesday in the morning. Then you have more time. In the classical protocol, you need to prepare on Friday to begin on Monday, or if you begin on Monday, you cannot make the tirage before Thursday. Then the idea, thank you Arnaud for this very nice new idea for the protocol. The idea was to shut and to delay the first step directly. We begin directly by the second step. We have seen that finally the yeast is capable to achieve in good conditions the first, the second fermentation, and why not to propose, because we have yeast very resistant, why not to propose directly a reduced, a shortened protocol. And as previously, we have to keep to follow correctly the evolution and the changes in the assimilable nitrogen. Then we have tried, as previously, to increase and to decrease by 30% the quantity. We see on the left part that we have just one step, and we have considered also the sugar, the residual sugar and the residual nitrogen at the end of the first step in the classical protocol. Then we have followed and integrated the 50% of sugar we had at the end of the first step. Then we can follow here the evolution, the changes in the sugar. We can see a correct and a continuous kinetics with the decrease of the sugar content. Then it's easy to see that we are after 2 days approximately at a bit lower than 50%. If we consider just 36 hours, we are approximately at 50% of the initial content in sugar. Then for the nitrogen, it's a bit different. We see on the left, after 2 days, we have no more nitrogen or DAP in the starter. Then it's necessary, because we have to work in 36 hours, it's necessary to follow, and it's not possible to reduce so much the DAP content. If not, we have no DAP in the starter preparation, and it can be a problem then to achieve correctly the second fermentation in the bottle. Then we need to keep the higher level and to follow to be sure that it's possible to continue in good conditions. Now, one of the more important parameters is the total population. We have followed the total population and also the live cells present in the starter. We can see that after 1 day and a half, 36 hours, we are between 60 and 80 millions of cells per milliliter. And for the classical value for the DAP, we are around 70 millions per milliliter. Then the quantity of yeast, the population is sufficient to inoculate with a low volume and to achieve correctly the prise de mousse in good conditions. We will see this just after. I will show you at the same time. Okay, here we compare finally the two different protocols. On the left part, you have the classical protocol in 3 days. We can see the decrease of the density because of the decrease of 50% of the initial sugar content, and after a second step, and we have a drop and a gap for the DAP content. For the new protocol proposed in 1 day and a half, it's a bit different. We have no first step, and we enter directly for the second step, and we adapt, of course, the DAP content. It's very essential to follow correctly the DAP content. If we compare now the two protocols, we can see the upper part of the graph is the classical, in 2 phases, 2 steps protocol, and the lower part is the new protocol. We can see that on the left for the sugar it's not so evident because we have a decrease, a continuous decrease of the sugar content, and it's the case for both protocols. But on the right we can see it's a bit different because we have the classical protocol and we see that we have a strong decrease in the DAP content between the first and the second phase, because we have initially 5 hectoliters, and for the second phase we have 30 hectoliters. We have a strong decrease. With the new protocol, we have the same shock for the yeast, and we have a continuous decrease, and we pay attention to be sure that after 36 hours we keep the quantity necessary to continue correctly. And it means that you begin on Monday afternoon, you are ready to begin the tirage on Wednesday in the morning, and you have the quantity of DAP necessary to continue the tirage all day long. Okay, then we follow the population, the yeast population. It's normal for the classical protocol. We see a strong decrease because we have a dilation from 1 to 6 volumes, and after we need more time to increase the population needed for the inoculation, for the secondary fermentation in the bottle. With the new protocol, we can see that we begin quickly with a high level of cells, and then we maintain this level. We have made it many times. We have not the so important increase during the starter preparation, and we can see the difference here. We are at 110 millions of cells per milliliter. Then it's very easy then to inoculate to achieve the secondary fermentation. Well, to conclude, 4 points. The first, and with the classical pied cuve, PDC, we need to prepare it in 3 days. Okay, and we have shown that it was possible to reduce and to keep an eye on the DAP content to be sure that we have no deficiency for the nitrogen. The first point. The second point, we have to optimize, of course, the protocol, and we have to pay attention to the temperature. Okay, it depends on the volume of the starter prepared, and then to follow the sugar is not so important, but the assimilable nitrogen. The third point is the performance of the yeast. We have seen that it was possible to achieve correctly the Prise De Mousse in the bottle. Sometimes, of course, if we have a fermentation at 13 or 12 degrees in some cellars, the case is longer. It can be 3 months. If we are at 16 or 17 degrees, it's shorter. It's always the same for the difference of temperature in the different cellars. And we have seen that finally we have pretty the same kinetics, and sometimes you are a bit quicker. Last point, because it's also very important, the testing notes. We have made the testing with the different winemakers, where we have made the trials, and with the colleagues from the laboratory of Reims. It's the university in the Champagne region, and we have seen that finally the profiles of the wine after the first fermentation and after a secondary fermentation corresponded to what was expected for this style of sparkling wine, the base wine and the sparkling wine. And we need, especially the freshness, we need, because it's what is expected by the consumers. Freshness and the notes from fruity nose and no evolution. This is very important, no evolution, because we will keep the wine during sometimes many years. And then the conclusion is that we hope that in the coming years it will be possible to use more and more this protocol to reduce the time and to continue the experiment to see how it's possible to convince you to use this strain and this protocol. Thank you for your attention.
Thank you very much. Muito obrigado. Não sei se tem alguma questão a colocar, ou Richard? No questions, so thank you very much.
Obrigado.
Thank you.
Thank you.